Structure of prokaryotes. There are many differences between prokaryotic and eukaryotic cells. However, all cells have four common structures. The plasma membrane, which functions as a barrier for the cell and separates the cell from its environment. The cytoplasm, a jelly-like substance inside the cell. Nucleic acids, the genetic material of the cell. And ribosomes, where protein synthesis takes place. Prokaryotes come in various shapes, but many fall into three categories: coxy, spherical, bacilli, rod-shaped, and spirally, spiral-shaped. Figure 1. The prokaryotic cell. Recall that prokaryotes, Figure 2, are unicellular organisms that lack organelles or other internal membrane-bound structures. Therefore, they do not have a nucleus but instead generally have a single chromosome, a piece of circular, double-stranded DNA located in an area of the cell called the nucleoid. Most prokaryotes have a cell wall outside the plasma membrane. Recall that prokaryotes are divided into two different domains: Bacteria and Archaea, which together with Eukarya, comprise the three domains of life. Figure 3. The composition of the cell wall differs significantly between the domains Bacteria and Archaea. The composition of their cell walls also differs from the eukaryotic cell walls found in plants, cellulose, or fungi and insects. Chitin. The cell wall functions as a protective layer, and it is responsible for the organism's shape. Some bacteria have an outer capsule outside the cell wall. Other structures are present in some prokaryotic species, but not in others. For example, the capsule found in some species enables the organism to attach to surfaces, protects it from dehydration and attack by phagocytic cells, and makes pathogens more resistant to our immune responses. Some species also have flagella, singular, flagellum, used for locomotion, and pili, singular, pilus, used for attachment to surfaces. Plasmids, which consist of extrachromosomal DNA, are also present in many species of bacteria and archaea. Characteristics of phyla of bacteria are described in Figure 4 and Figure 5. Archaea are described in Figure 6. The plasma membrane. The plasma membrane is a thin lipid bilayer, 6 to 8 nanometers, that completely surrounds the cell and separates the inside from the outside. Its selectively permeable nature keeps ions, proteins, and other molecules within the cell and prevents them from diffusing into the extracellular environment, while other molecules may move through the membrane. Recall that the general structure of a cell membrane is a phospholipid bilayer composed of two layers of lipid molecules. In archaeal cell membranes, isoprene, phytanyl chains linked to glycerol replace the fatty acids linked to glycerol in bacterial membranes. Some archaeal membranes are lipid monolayers instead of bilayers. Figure 6. The cell wall. The cytoplasm of prokaryotic cells has a high concentration of dissolved solutes. Therefore, the osmotic pressure within the cell is relatively high. The cell wall is a protective layer that surrounds some cells and gives them shape and rigidity. It is located outside the cell membrane and prevents osmotic lysis, bursting due to increasing volume. The chemical composition of the cell walls varies between archaea and bacteria, and also varies between bacterial species. Bacterial cell walls contain peptidoglycan, composed of polysaccharide chains that are cross-linked by unusual peptides containing both L and D amino acids, including D-glutamic acid and D-alanine. Proteins normally have only L amino acids. As a consequence, many of our antibiotics work by mimicking D amino acids and therefore have specific effects on bacterial cell wall development. There are more than 100 different forms of peptidoglycan. S layer, surface layer, Proteins are also present on the outside of cell walls of both Archaea and Bacteria. Bacteria are divided into two major groups, gram-positive and gram-negative, based on their reaction to gram staining. Note that all gram-positive bacteria belong to one phylum. Bacteria in the other phyla, Proteobacteria, Chlamydias, spirochetes, cyanobacteria, and others, are gram-negative. The gram staining method is named after its inventor, Danish scientist Hans Christian Gram, 1853 to 1938. The different bacterial responses to the staining procedure are ultimately due to cell wall structure. Gram-positive organisms typically lack the outer membrane found in gram-negative organisms. Figure 7. Up to 90% of the cell wall in gram-positive bacteria is composed of peptidoglycan, and most of the rest is composed of acidic substances called teichoic acids. Teichoic acids may be covalently linked to lipids in the plasma membrane to form lipoteichoic acids. Lipoteichoic acids anchor the cell wall to the cell membrane. Gram-negative bacteria have a relatively thin cell wall composed of a few layers of peptidoglycan. Only 10% of the total cell wall, surrounded by an outer envelope containing lipopolysaccharides, LPS, and lipoproteins. This outer envelope is sometimes referred to as a second lipid bilayer. The chemistry of this outer envelope is very different, however, from that of the typical lipid bilayer that forms plasma membranes. Archaean cell walls do not have peptidoglycan. There are 4 different types of Archaean cell walls. One type is composed of pseudopeptidoglycan, which is similar to peptidoglycan in morphology but contains different sugars in the polysaccharide chain. The other 3 types of cell walls are composed of polysaccharides, glycoproteins, or pure protein. Reproduction. Reproduction in prokaryotes is asexual and usually takes place by binary fission. Recall that the DNA of a prokaryote exists as a single circular chromosome. Prokaryotes do not undergo mitosis. Rather, the chromosome is replicated and the 2 resulting copies separate from one another. Due to the growth of the cell, the prokaryote, now enlarged, is pinched inward at its equator and the 2 resulting cells, which are clones, separate. Binary fission does not provide an opportunity for genetic recombination or genetic diversity. But prokaryotes can share genes by 3 other mechanisms. In transformation, the prokaryote takes in DNA found in its environment that is shed by other prokaryotes. If a non-pathogenic bacterium takes up DNA for a toxin gene from a pathogen and incorporates the new DNA into its own chromosome, it too may become pathogenic. In transduction, bacteriophages, the viruses that infect bacteria, sometimes also move short pieces of chromosomal DNA from one bacterium to another. Transduction results in a recombinant organism. Archaea are not affected by bacteriophages but instead have their own viruses that translocate genetic material from one individual to another. In conjugation, DNA is transferred from one prokaryote to another by means of a pilus, which brings the organisms into contact with one another. The DNA transferred can be in the form of a plasmid or as a hybrid, containing both plasmid and chromosomal DNA. These 3 processes of DNA exchange are shown in Figure 9. Reproduction can be very rapid, a few minutes for some species. This short generation time coupled with mechanisms of genetic recombination and high rates of mutation result in the rapid evolution of prokaryotes, allowing them to respond to environmental changes, such as the introduction of an antibiotic, very quickly. Evolution connection. The evolution of prokaryotes. How do scientists answer questions about the evolution of prokaryotes? Unlike with animals, artifacts in the fossil record of prokaryotes offer very little information. Fossils of ancient prokaryotes look like tiny bubbles in rock. Some scientists turn to genetics and to the principle of the molecular clock, which holds that the more recently two species have diverged, the more similar their genes, and thus proteins, will be. Conversely, species that diverged long ago will have more genes that are dissimilar. Scientists at the NASA Astrobiology Institute and at the European Molecular Biology Laboratory collaborated to analyze the molecular evolution of 32 specific proteins common to 72 species of prokaryotes. The model they derived from their data indicates that three important groups of bacteria, actinobacteria, Deinococcus, and cyanobacteria, which the authors call pterobacteria, were the first to colonize land. Recall that Deinococcus is a genus of prokaryota bacterium that is highly resistant to ionizing radiation. Cyanobacteria are photosynthesizers, while actinobacteria are a group of very common bacteria that include species important in decomposition of organic wastes. The timelines of divergence suggest that bacteria, members of the domain Bacteria, diverged from common ancestral species between 2.5 and 3.2 billion years ago, whereas Archaea diverged earlier, between 3.1 and 4.1 billion years ago. Eukarya later diverged off the Archaean line. The work further suggests that stromatolites that formed prior to the advent of cyanobacteria, about 2.6 billion years ago, photosynthesized in an anoxic environment, and that because of the modifications of the pterobacteria for land resistance to drying and the possession of compounds that protect the organism from excess light, photosynthesis using oxygen may be closely linked to adaptations to survive on land. Section summary. Prokaryotes. Domains Archaea and Bacteria are single-celled organisms lacking a nucleus. They have a single piece of circular DNA in the nucleoid area of the cell. Most prokaryotes have a cell wall that lies outside the boundary of the plasma membrane. Some prokaryotes may have additional structures such as a capsule, flagella, and pili. Bacteria and archaea differ in the lipid composition of their cell membranes and the characteristics of the cell wall. In archaeal membranes, phytanyl units, rather than fatty acids, are linked to glycerol. Some archaeal membranes are lipid monolayers instead of bilayers. The cell wall is located outside the cell membrane and prevents osmotic lysis. The chemical composition of cell walls varies between species. Bacterial cell walls contain peptidoglycan. Archaean cell walls do not have peptidoglycan, but they may have pseudopeptidoglycan, polysaccharides, glycoproteins, or protein-based cell walls. Bacteria can be divided into two major groups, gram-positive and gram-negative, based on the gram-stain reaction. Gram-positive organisms have a thick cell wall, together with teichoic acids. Gram-negative organisms have a thin cell wall and an outer envelope containing lipopolysaccharides and lipoproteins. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.